So I want to move on to initiation of replication. Right? So how do we get these replication forks? Where do they come from? And what are the proteins that are involved in this process? And what I want to do first is to go through the process of initiation in E. coli, at the E. coli origin of replication. So I've already told you that the E. coli origin has these two sequences, the 13-mer and the 9-mer. And the 9-mers are what are recognized by the initiator protein. And not surprisingly, given its name, the initiator protein is the first protein to act at an origin of replication. Okay? It binds the 9-mer sequences here. It does so using a double-stranded DNA binding domain that's located at its C terminus. When it does this, it, in the presence of ATP, it has to be bound to ATP to do this. It actually drives the unwinding of the DNA at the 13-mer region of the origin. Okay? It does this via a single-stranded DNA binding domain that is separate from its double-stranded DNA binding domain, and it actually forms this remarkable helical form of the protein, binds to the single-stranded DNA. And it binds to one strand, leaving the other strand available to be bound by other proteins. Okay? So unwinding occurs with just DNA A and ATP. You mix these two things together with a circular form of either the chromosome or a plasmid, and this works fine. It does need to be negatively supercoiled for this to work. Okay? Remember how we talked about the E. coli genome is kept by gyrase in a negatively supercoiled state? It turns out if you use this on just a straight piece of linear DNA, you have to have a lot more DNA A to make this work. If you have it on a supercoiled plasmid, it works actually quite nicely with just a modicum of DNA A. Okay, so that suggests that it's using some of that negative superhelicity, negative superhelical energy stored in the DNA to drive the unwinding event. Okay, and recall these 13-mers are actually very AT-rich. Okay, or actually, I'm not sure I said that, but they are. So that also means it's easier to unwind. Okay, so what happens next? So the next step is I told you about this DNA B being the helicase, and we just briefly mentioned this protein DNA C. Okay? DNA B and DNA C form a complex off the DNA. And they have two interesting properties when they're together. One is that DNA C turns off DNA B. Okay? So DNA C is an inhibitor of DNA B. Okay? And the second is what's illustrated here, that DNA C and DNA B, this complex, interacts with the bound DNA A and opens the DNA B ring and places it around the single-stranded DNA. Okay? And what's more remarkable about this is that on the strand that DNA A binds, it puts it on in one orientation, and on the opposite strand, it puts it on in the opposite orientation. Okay? We know there are interactions between A and C that are necessary for this, as well as interactions between A and B. It's still entirely unclear how you get these two opposite orientations on the different strands. Okay? So not everything is known yet, even though people have been studying this for a long time. Okay, so at this point, you have two helicases loaded around single-stranded DNA at the origin of replication, facing opposite directions. Okay? And they're basically facing the directions they need to go in order to drive their individual replication forks. One helicase is going to be required for the replication fork going to the right, and one will be involved in the replication fork going to the left. Okay? Interestingly, one other kind of interesting thing is they're actually loaded such that the one that goes to the right is initially on the other side of the one that goes to the left. So they actually pass each other as they start unwinding. So there's a problem, though, is at this point, they're not unwinding anything, right? So why is that? DNA C is still bound. Okay? So they're around the single-stranded DNA, but they're not active. So the question is, what activates them? And this is a nice way of making sure that the way the cell does this makes sure that the priming has initiated before the helicase has been activated. Okay? And that is that synthesizing an RNA primer, and again, the direct mechanism to understand this is not clear, but when you synthesize an RNA primer, DNA B and DNA C separate, and DNA C is basically released into solution. It's actually not a hexamer, really. It's actually disintegrates in the absence of DNA B into monomers. Okay? And then the helicase can start moving. Okay? When it's not bound to DNA C, it's able to become active. Okay? Everything from here is downhill, right? We've got a primer, and this is our leading strand primer. I know we talked a lot about priming. It was always about the lagging strand, but in fact, the leading strand needs a primer as well. And what you'll see is the first leading strand primer is made in very much the same way as the lagging strand primers are. Okay? And that'll become clear as we go along. But we have a primer here. So now the sliding clamp loader, okay, associated with the 3 polymerases, can recognize that primer. Okay? So it binds the primer, puts a sliding clamp around each of the primer template junctions, around the double-stranded DNA regions. Okay? And at that point, that can be recognized by one of the DNA polymerases. Okay? We have now an engaged polymerase elongating one of the 2 primers. There's another one going this way. Okay? So note that the bottom strand is priming for this direction, the top strand is priming for the other direction. They're made by the helicase that's going the opposite direction. Okay? So that's just like how the Okazaki fragments are primed, right? The primer is always made so it's priming the synthesis going the opposite direction of the helicase. Okay? So one more time, let me go back. This primer was made by the helicase going this direction. Okay? But it will be extended by the polymerase that associates going the other direction. Okay? So it's really the same process. This helicase primes this leading strand, and it's as if it's one of its Okazaki fragments. Okay? Because it will prime the Okazaki fragments in this region, and the opposite is true on the bottom strand. Okay? Now what happens? Well, you keep making your leading strand. Primase rebinds, puts another primer on here. A second polymerase engages. Okay, after a sliding clamp is put on, it starts extending. And then just as we talked about before, a third primer will be put on such that a third polymerase can be engaged in the lagging strand. And then when this one finishes, this polymerase will be released, and a new primer will be presented, and you have your 3 polymerase reaction going. Okay? So the last part is really what we were talking about before. It's just now we see the initiation step, that first leading strand synthesis being initiated, as opposed to before we always showed it as already ongoing. So are there questions about this?
How does sliding clamp eventually assist?
Sliding clamps actually are taken off when they're not occupied by proteins. Okay? So it turns out that it's much easier to take off a sliding clamp than it is to put one on. And you can think of it most easily as an entropic issue. Right? To take it off is entropically favorable. You're removing it from the DNA. So actually there's extra delta prime in the cell. Okay? The amount of delta prime in the cell is in excess to the amount of the clamp loader. And delta prime all by itself can pop the clamps open. And when they pop open, they just fall right off the DNA. Okay? So why are they protected? Right? And this is actually quite clever. So the place that delta prime binds is exactly the same place that the polymerase binds. So if the polymerase is binding to a clamp, delta prime can't act on it. Okay? And that actually turns out to be true for any type of protein interacting with a sliding clamp. And this is true in eukaryotic cells. It's true in prokaryotic cells. An occupied clamp is not a substrate to be removed. And you'll see more, not so much in E. coli, although, no, we'll talk about it in E. coli for repair. There are DNA repair proteins that use the sliding clamp. Okay? And if they're bound, the clamp stays on even if the polymerase is still is done with it. Okay? And that turns out to be involved in mismatch repair, which we'll talk about in a week or two. Okay? Other questions? You've had one. You don't have it anymore.
Well, I kind of do, but I kind of just make sense of it.
Okay. How many sequence-specific DNA binding proteins are required to do this? 1. Right? DNA is the only sequence-specific binding protein required to assemble this big, huge machine. Everything else is either a protein-protein interaction, for example, between DNA A and DNA C and B, or a non-specific protein-DNA interaction. Okay? The helicase has affinity for single-stranded DNA, but it's not a sequence-specific interaction. Okay? The polymerase has affinity for a primer-template junction, particularly one that has a sliding clamp, but it's not sequence-specific. Okay? That's driven by non-specific or structure-specific interactions. And it's worth keeping in mind that you can do quite a bit with only a little bit of sequence-specific information. Right? You can build this big, huge, if DNA were a meter wide, size of a FedEx truck machine, by starting with just one little, I think DNA A would probably be the size of a beach ball or something in that sort of world. Okay? Everything else is sort of recognizing or working by structure recognition rather than sequence recognition. Okay? And while this is true in replication, what you'll find is it's also true in transcription. It's also true in translation. There's a lot of things that recognize structures rather than sequences, and that's worth keeping in mind.